Great. Greetings, I am Whitney Spencer, of a junior biology major of Alabama A&M University, and as an NSF REU student this summer, I've been working in Dr. Brandi Kiel's Reese lab, and today I present to you my research on monitoring subsurface fungal growth in the South Atlantic Transit. Just as an introduction, fungi are a very diverse and essential group. They make up about almost 80% of Earth's biofungal mass, and they're capable of nutrient cycling, interaction with plants and animals, and even decomposition. With my study, we had 2 fungal isolates that were collected from the South Atlantic Transit, which is in the South Atlantic Ocean right off the coast of Brazil, with SATF14, which is a species of fusarium. It was collected at about 90 meters below seafloor, and SATF24, which is a species of cladosporium, which is collected at about 106 meters below the seafloor. And with these 2 isolates, our objective of this study was to just learn how subsurface fungal growth is affected by environmental characteristics. And we hypothesized that at about 24 hours of growth, that we will be able to see the growth, and at about 400 hours, the growth rate will decrease. And our methodology for this experiment goes as such. We streak plated both of the isolates on 25% potato dado extras in order to induce spore formation. After we put those in the incubator, they stayed in there about 7 days, and after we noticed spore formation, we extracted those spores using 0.2% SDS solution. And after spore extraction, we diluted them using serial dilutions and started spore counts. After spore counting, we calculated how many spores we needed to put into every vial in order to start our growth experiment so that it would be an equal growth. And after we did that, which was about 1 times 10 to the 4th power amount of spores, our growth experiment was started. And with our growth experiment, we achieved these results. And how we achieved these results, previous to filtering the fungi through the filters, we weighed the filter beforehand, and after filtering and allowing them to stay in the drying oven for about 18 hours at about 80 degrees Celsius, we took them out and took the final weight. And the subtracted weight is what gave us these results. And as you can see, we had 3 samples, which consisted of a control in the orange, STF14 in the blue, and STF24 in the green. And as you can see, these results created a fungal growth curve in which you can see the fungi going through their entire life phase up into the stationary phase. And with these results, we concluded that SATF14 had about a growth rate of 1.20 times 10 to the negative 2nd power milligrams per hour, and that SATF24 had a growth rate of about 7.85 times 10 to the negative 2nd power milligrams per hour. And based on these results and further literature, we concluded that these species of fungi that have been closely related to other species found in further literature, that they do line up with their alike species. Based on SATF14, we noticed that it does line up with another fusarium species that we found in further literature, and that SATF24 also similarly aligns to another cladosporium species, but due to difference in metrics and design and experimental design, it's kind of hard to make those comparisons. And in the end, based on our hypotheses, hypothesis A, which stated that about 24 hours we start to see fungal growth, our hypothesis wasn't exactly wrong, but we hypothesized a bit too much. At about 24 hours of growth for both isolates, SATF14, which is in the blue, was still within this lag phase. And SATF24 at about 24 hours was still in this lag phase as well. And hypothesis B states that at about 400 hours, we will start to see a decrease in fungal growth. And that hypothesis was a bit overdone as well. As you can see, at about 90 hours, SATF14 stops its growth, decreases its growth, and at about 190 hours, SATF24 decreases its growth as well. And our overall objective of this experiment was to just get a fungal growth curve, and in the end, it was achieved. Future work consists of testing these fungi in order to see their effectiveness on different other types of characteristics, such as pH, temperature, and salinity. Thank you.